that are still and she lives on the farm.
So just a heads up that we are recording as you speak. You might be captured.
You might be captured. There's a chance. There's a chance. I can do urban, I can do urban. Yeah.
Okay, everyone, it is time, so let's get started. So, welcome everyone to the 2023 of the Biology Meet and Greet. I believe this is a 237th edition? 238th. 238th edition of the Bio Meet and Greet. Long tradition in the biology department. I'm kidding. Confused. But thank you for coming. So what we're going to do is have a little bit of a get-together so you can hear about the things that are going on in the biology department, hear from some of the faculty that are conducting research and doing the teaching, and you'll also hear a little bit about the clubs and the affinity groups that are available as a resource in the universities. So I'm going to start by giving the floor to, well, let me make an announcement to the speakers. So we have a little slide forward projector, so you can just click the button and it will move forward. So I will give the floor to our chair, Dr. Timothy Sparks.
Well, it's a busy slide, but I promise I'm not going to spend too much time away from anybody else. But if you don't know me, I'm Dr. Sparks. I've got a strange accent. I'm from Wales originally in the UK, went to London for my undergrad, University of Kentucky in Lexington, Kentucky for 11 years of my life. So I say y'all in between British things. It's all mixed together. I'm a first generation student. I was also an international student as a grad student. So I've got a real good feel, at least my own experience of the challenges that people face going through. And a lot of what I do as chair is often connected to the themes of trying to provide those support systems and resources for all of you. We talk a lot about the ways to be successful, many programs that we have available to you. The general theme is we're here to support you, provide a diversity of options that will allow you to have a diversity of career options for yourself and help as best we can to get you there, empower you to have the confidence to navigate your journey yourself. So we're not telling you what to do, we're helping you get to the place where you can know for yourselves what to do. So I'm going to show you a couple of slides, and the theme is not for you to look through all these slides. They're all posters in the hallway, so just kind of go look at the posters afterwards. But a couple of things people don't know much about. People often come into the BS, but there's also a BA. So if you want to know more about the BS BA, there's stuff on the poster. We can talk to you more about it as well. Some other things. A big thing for us, and it's in the mission statement, we'll read the slide on that side, is trying to provide, again, a diversity of course options, a diversity of career options, and a diversity of authentic research experiences, which we all found, we personally think are very important to development. There's lots of research talking about the importance of research exposure for students' development, and we have loads of options. So again, the theme is not to go through every one of these, but we've got these pure courses, a bunch of them, research, immersion, a bunch of students here I know have taken some of these courses. We've got mentored research courses, we've got experiential learning courses, which you're required to take that can be done through research, funding, all kinds of funding around student research, all kinds of Dean's undergrad fellowships, additional funding. So lots of funding, lots of research options through coursework or through individual faculty members. It's more about finding what's a good fit for you, learning the right time to do it, approaching professors appropriately when you're ready for those kinds of potential moves or courses, and then stepping into that space to get the most from it. Loads of evidence that that's hugely beneficial for what you want to do later, even if it's not directly in research. So just one thing to touch there, that's why I'm showing the slide. One other thing is just where these things end up. So there's lots of these outcomes of all these experiences, lots of these things at the top about students graduating from the program. Clubs, again, are going to come up later more. So clubs to get involved with, awards, everything you can imagine is here to provide you with the diversity of options. It's just a matter of you stepping into it. And the last one just to kind of bring up is just all this stuff is on the way. I did the CSA's Fest yesterday. One of the most common, a couple of common things. One is we've got a beekeeping course on the way, and Bill's got some bees here for you today. Lots of interest in the beekeeping course. And then there's 3 plus courses, so there's 3 plus programs. So they're on the way. We're working on them right now. There's 5 coming. Potentially that's 4. So again, over all the details, they're all on the posters outside. I just want to kind of show you the range of things that are coming. The biggest thing I want to leave you with is I teach capstone, which is with the senior students. I always ask them, if you could go back and be a freshman again, what would you do differently? One of the most common things they say is take advantage of the resources that are available to them, take advantage of the support. So all the support you could ever need is here. We got you. You just got to jump in to take advantage of the support. So jump in, take advantage, and we're here to guide you and support you and help you build confidence for the rest of your time and beyond. So welcome. Looking forward to all of the other speakers. I'll see you again when I talk about my stuff later. All right.
Okay, so now we will have some of the faculty, the research faculty, will start telling us a little bit about their research. And so we're going alphabetically, so I'll start. I'm Windsor Aguirre. Yeah, this is, Christian is very adaptable. He's a graduate student in the program, very adaptable. Amazing what he can do. Anyway, so I'm an evolutionary biologist, and I'm interested in understanding how organisms diversify and adapt to nature. I'm particularly interested in the relationship between the environment and the form of the organism. So we work with freshwater fish primarily. Some of the work is morphological. We try to understand how body form, why it changes in the ways it does, and how that matters. And then some of the work is using DNA sequencing. And so we also study, we use DNA sequences as markers of evolutionary history and gene flow. I'm particularly interested in trying to bring in new technology. So one of the things we're doing is bringing in a new type of relatively new type of sequencer that generates genome level data in real time and can be used in a classroom setting. So you can, if you take classes, if you take evolution, if you take molecular methods with me, you'll probably be exposed to it and see it working. So there's some great technological developments that are rising. I teach evolution, our sophomore level class, Bio 235, Biostatistics, Bio 206. And I would encourage you to try to take that as early as you can in your career. It is an important class for the higher level classes. This spring, I'm going to be teaching molecular methods in ecology and evolution, in which students, we do a little bit of training up front, but then students design their own research projects in groups. We vote on those, and then you actually go about collecting the data and collecting the specimens, collecting the data, and analyzing the data, and using that for a report. So it's a research-based course that employs molecular methods, and especially DNA sequencing. I have a non-majors class in evolution, health, and medicine. You might not link evolution with health and medicine with your health. Big mistake. You have an evolutionary history, and it impacts your health in many, many, many ways. Ways that we're beginning to understand, and that are helping us establish better policies to manage many of the large-scale health problems that we're having. Concepts in evolution, an upper-level evolution class. General biology, I'm not teaching this year, but I've taught in the past. And if you're a freshman, this year in the winter quarter, there's a focal point seminar that's a required course for freshmen at DePaul. But this is a study abroad variant. In this study abroad variant, we meet for classes in the winter quarter like a normal seminar. And then in the spring break, we head to Ecuador and we learn about the other phase, the other side of being a biology student in 2023. The positive side is you have all these incredible advances in technology. As undergraduates, you can do things many of your faculty, including myself, probably couldn't imagine were possible to do as a student. You have access to extraordinary technology that gives you tremendous power in trying to understand the natural world. But you also live in a time in which the environment is in worse condition than it ever has been. And tomorrow, literally tomorrow, that's 23rd of September, it's going to be worse than today. And the 24th, I'm going to bet it's going to be worse than tomorrow. There's 8 billion people on Earth, having enormous consequences on every ecosystem. And so that is a reality. In this course, we talk about some of the causes of this extinction crisis. And we survey natural ecosystems. So we go to the high Andes Mountains, see some extraordinary creatures there like Andean condors. We also look at the large-scale deforestation, the melting of glaciers in the mountains that are a source of water for people in the mountains. We go to the rainforest, the headwaters of the Amazon forest. We go to stay with a native village. They have an eco-lodge, not luxurious, but plenty of snakes and spiders. That doesn't tell you, I don't know what will. And we spend a few days there with the community and talk about the problems that are affecting rainforests throughout the Earth, including in the Amazon basin, in that region, one of the most biodiverse ecosystems on Earth, with one of the largest oil deposits underneath. Huge source of tension about how to exploit that oil, but not destroy the rainforest. And then we finish, as we should, with a trip to the beach. We go to the coast, and we visit a preserve and talk about some of the threats like ocean acidification and plastics. Okay, so deadline is November 1st, so if you're interested, make sure to check it out. I'm glad to answer questions about it, but I hope maybe I'll see a couple of you there. And there is it's a study abroad course, so there's a fee, but there is financial aid. So don't not apply because there's a cost. You might get a scholarship. Okay, one more thing I forgot. So my lab, we do research on the freshwater fishes of Western Ecuador. A lot of the literature is in Spanish, especially the details about natural history. And I'm trying to publish in Spanish. The problem is, it takes me a million years to do anything these days. So it'd be great to have some students that can help reading that primary literature in Spanish and help translate some of the work that we're doing in English so we can publish in Spanish. So if you speak Spanish or have interest in computational methods, also because of nanopore sequencing, enormous data sets, please come talk to me. Okay. With that, unfortunately, Dr. Bell could not be here today. She meant to be here, but she had a parental emergency. And so she is teaching these classes: cell biology, physiology of poverty, which I understand is incredibly popular. I would have loved to take that class. Brain and behavior, endocrinology, introduction to health. So she teaches courses in health sciences and in biology, besides biology. And her research is on neurodevelopment, endocrinology, immunology, and toxicology. So one of those fields probably interests you. So check out her lab. Her webpage is here, and you can take a quick picture if you wanted to check out her webpage. And I'm going to copy this for next year. Good idea. 1, 2 seconds. We got it. Okay, Dr. Brooks.
Hi everyone, good to see the turnout today. So welcome to Fall Quarter. I hope it's going well for everybody. So my name is Dr. Brooke. For those of you that don't know me, I am Canadian and I'm trained as a microbiologist. And up here, I realize there's a couple of errors on the slide, so I'm going to correct that right now. I'm teaching microbiology Bio 210 in the fall. And in winter quarter, I will be teaching advanced microbiology, a secure course with Dr. Scrimenti. And in the spring, I will be teaching Bio 347, which should be topics in medical bacteriology. So as you can hear, my interests lie heavily in the area of infectious disease. I want to mention the Cure course for a minute. We launched this last year, and we're having some nice feedback from students about their experience, and I highly recommend it. If you have taken 210 or about to take biology, microbiology 210 and find you really like it, consider taking advanced microbiology. You get to design your own independent projects and carry them out. And we are hoping to put together enough data to submit to DePaul Discoveries pretty soon. So it's exciting. Let's see, that's my contact information. In terms of my research, as I said, I'm a microbiologist, so my area is infectious disease. I have a graduate student with me in the lab at the moment, and I have several undergraduates working with me. And I really like having students in the lab. I like the interaction, and I like my students to be involved in all aspects of the research. So what we primarily focus on is this bacterium up here, which is Stenotrophomonas multifida. It's an infectious bacterium that causes primarily respiratory tract, bloodstream infections, and it's particularly significant in those patients that are susceptible to it, namely immunocompromised patients. So this can be a cause of a fatal infection in an immunocompromised patient. So one of the things we're looking at in my lab is to understand the molecular mechanisms that underlie this organism's ability to persist and survive in the environment and then also in the patient. This bacterium has the ability to form what are called bacterial films or biofilms. And biofilms, for those of you who may have heard the term, these can act as sources of infection. And so one of the things we're looking at, actively involved in, is to try to find different ways to treat and prevent the formation of a biofilm. So reduce the incidence of infection, perhaps, in patients. The other thing I want to mention about this bacterium is it's multi-drug resistant. And we are looking for new ways to treat infections by this organism. It is limited in the number of different antibiotics that are effective against it. So one of the things we're doing is exploring new pathways to get around even using antibiotics. Let's try something else altogether. And so my undergraduates are involved with me, along with my graduate student here in the audience, Sophia right there. And we're looking at different ways to try to prevent infection by this organism. So this is me in a nutshell. And I just put this slide up here because I want to say to you, I understand the importance of undergraduate research experience. I was, like many of you, an undergraduate who in my third year was searching for that experience. How do I get it? And I found it intimidating somewhat to approach a professor, and he turned out to be very, very accommodating, very nice, welcomed me into the lab, even insisted I call him by his first name. And it was a great experience. From that, I learned, okay, this is what I really want to do. And I think I might actually be good at it. And so getting the experience early, I can't encourage it enough. So if you're at all interested in infectious disease, come talk to me. I always have a number of students who are asking to get into the lab. So I hope to see you. In terms of publications, yes, you can see that we have publications up here with undergraduate co-authors. So I published just with undergraduate students. And it's a big challenge for me. It's a challenge for the undergraduate students, but I know they're going to get a lot out of it. They're involved in all steps of the process, from the experimental design to carrying out the experiments to getting the data, doing the analysis, and writing up the paper. So you're getting the whole thing. Again, we've had several poster presentations at different types of conferences, at national, international, and local. So again, you've got my contact information, jbrook@depaul.edu. If you're interested in what I do, contact me by email, and we will talk. There's specialized training you have to go through, but I'm always welcome to have that discussion with you. Okay, with that, I'm going to pass it to Dr. Bystriansky, who's at the back.
passing the baton to
another person that went to the same school and is from the same country. Isn't that crazy that that happened? So I went to Guelph along with Dr. Brooke, slightly different time, different department. I did an undergrad in marine biology, Bachelor of Science, and then went on to do my PhD in what's called comparative animal physiology. Essentially, I'm interested in how animals work, how the body works, especially when that animal is dealing with either fluctuating or changing environmental conditions. So Dr. Aguirre gave a great, really uplifting, example of what's going on in the world today, and all of the organisms out there are dealing with that too. And so I'm interested in how they're able to hopefully survive and what adaptations they might have to deal with those types of scenarios. So a bit about my teaching. I teach Gen Bio 3, usually with Dr. Dean. Gen Bio 3 is essentially the biodiversity and physiology of plants and animals. So if I haven't seen you already, I'll see you maybe in the spring. I also teach a non-majors marine biology course, and most of my courses are obviously in the physiology area. So I teach animal physiology third year, animal phys, and advanced comparative physiology, which is essentially environmental physiology, how our animals adapted to live in different environments. I also teach a study abroad program where we take students to Spain every summer. And this is a program where essentially students learn how to do research. So we kind of think of it as sort of research boot camp. We're going to spend a few weeks coming up with project ideas, planning experiments, traveling to a foreign country where I can't speak Spanish, which is really interesting, and then trying to pull off an experiment over a 2-week period. And so we've been really successful in this program. I teach it with Dr. Bryce in chemistry, so we take students from all different science majors. And it's one of our examples of a cure here, a course-based undergraduate research experience. So if you're interested in getting research experience, but you haven't broken in and gotten to the lab yet, maybe this is a way to do that. This quarter, I actually started teaching Bio 307 as a cure as well. So the entire lab portion of animal physiology is like an open, research-based, project-based course. And so this is the first year I'm piloting this idea. So far, so good. Hopefully, we're going to have some really interesting experiments coming out of that, and some things to share maybe at the end of the quarter with the wider community. So my background for my research, here we go. As I mentioned, I'm a physiologist, and I grew up, I loved fishing as a kid. One of my sort of passions was sport fishing. And I learned that I love fishing for salmon and trout. And so I just got engaged in the life history of these amazing fishes. And so luckily, I was able to take my childhood passion and turn it into a career. I look at how salmon and different species within the family are able to go through this amazing life cycle where they're transitioning from life as a freshwater fish to one as a marine fish. And if you imagine the last time you got a gulp of seawater, how uncomfortable that was, all marine fish are drinking seawater all the time. And somehow their physiology allows them to survive in those conditions. So I study how a freshwater fish can transition into a marine fish and back again, all as part of their natural life cycle. I've extended this beyond salmon over the last few years and started looking at other fishes as well. Essentially, this concept of what we call uric-salinity, the ability of an animal to tolerate changes in salinity, is the main area of my research. So if you're interested in this or any question related to an animal and its ability to deal with harsh or changing environmental conditions, I welcome you to email me or see me in the hallway and chat about possible projects and research opportunities. I've always got openings in my lab that anybody who's interested wants to talk to me about. All right, so that's me. Next up is Dr. Khan. Oh, all right, Dr. Khan. Good to welcome you.
Yes. Hi, everybody. So I'm Dr. Andrew Conniff. I am a new faculty hire in the department here. So I'm kind of learning everything at the same time that you all are. And so some of the main classes that I'll be teaching in the next year or so, some of you may actually recognize me from from some of the Gen Bio series in the labs and things. But then I'll also be doing biostats as well. And so this is a big passion of mine, trying to be quantitative, trying to understand how to bring stats and trying to understand how quantitative methods can all work. And then one of the big things that I'll be doing is this developmental biology course. And so I do hope that maybe I can try to design one of these cure-based courses, these more sort of research-intensive courses, to give you a sense about how developmental data is collected, processed, and then potentially even published as well. And so this will be coming in the next academic year. And so what do I actually do in my lab? So I am interested in heads, the shape of heads. So the main organism that I use mostly is cichlids. So you can kind of get a sense from these fish here that they look wildly different despite all being in the same family. And this is something that's kind of been puzzling me for a long time. Like, how do you get something that looks like this and looks like this in the same family? And so I try to employ a number of different methods, so molecular methods, developmental methods. I also try to go, try to take the 1,000 to 30,000 foot view as well, so looking across a whole host of different species as well. And so I'm basically interested in how the head develops. So the head is this wildly multi-complex structure. It can communicate, it can feed, it has sense organs like our brain and our eyes. And so there's all these different kind of competing features that are going on, but they all have to work at the same time. And they all have to be able to allow the fish to survive. And so a lot of my work tries to understand how they do this. So from both the embryo all the way up to the adult stage. And so I look at bones. This is the neurocranium, so this is kind of the skull of a cichlid. And then I also look at muscles. So this is a stained, CT scanned cichlid. And so there's the lenses right there. It's kind of very spooky looking right at you. And then another aspect of my work is to try to understand this transition from genotype to phenotype. So I'm interested in form, but I'm also interested in how we get those different forms. This is my lab right now. I tried to put like a little tumbleweed on it, and it basically just looked like a piece of trash hiding in the corner. So there's only myself and Sarah, my graduate student right now. And so I have lots of space for people who want to come in and make this lab look a little bit messier. Because it's very clean right now. Safe, but messy. And so if some of this stuff kind of interests you, feel free to contact me here. And this is my website here. I need to get one of those fancy little QR codes. I think you can also just Google my name, and I should show up. And I'm also on the faculty pages. So if you want any more information, like publications, but some sort of other cool images of fish and such, then take a look at my website to learn a little bit more about what I do. Okay. And then next up is Sarah. Oh, Dr. Connor.
All right. I'm Sarah Connolly. I have a joint appointment in health sciences. In biology, I teach intro bio. I'll be teaching that next quarter, so if you're not taking it now, maybe I'll see you next quarter. In other years, I've taught Bio 210 microbiology. I've also taught in the past a molecular virology course that bio students got credit for. And then I teach a course for public health students in the health sciences major, so that may or may not, probably doesn't include many of you. I study herpes viruses. I've actually calculated as I sat here that if this marks 25 years of me studying the entry of viruses into cells. I started this at the beginning of graduate school. So like 25 years of figuring out how, I don't know what the button is. That's cute. This turns into that. So this is an electron micrograph of a herpes virus particle. The DNA for the virus is in this little shell. It's surrounded by a membrane. And the virus binds to a cell, and then it fuses that membrane with the cell as the first step of infection, the entry of the virus into the cell. And that's all I study, is how do you get from here to here? But I use cell and molecular approaches, some virology approaches, and some protein biochemistry to understand how this happens. It's not a spontaneous event. It requires some proteins to interact with each other in a specific manner and to physically fuse those two membranes together. So I'll give a little pitch on viruses and why I find them interesting and fun to study. So these have really small genomes. So you can have viruses that have only a dozen genes. And yet that's enough for the virus to spread through a population. And as we've seen, around the world, they have a genome, it's protected by some protein. And then depending on the virus, it may or may not have a lipid layer around that. For herpes, it does have that lipid layer that you just saw. But that's it. There's no metabolism, there's no mitochondria, there's no protein translation happening in that little virus particle. But they infect our cells and then they take over the life of our cells. So this is a little image of the replication cycle of a virus. The first step is binding to a virus, binding to a cell. Entering that cell, the genome gets dropped off, it gets replicated by the cell, viral proteins get made, little virus particles are assembled, and then they bud out of the cell depending on the virus. This can take, for some bacteriophage, these are viruses that infect bacteria, that can take 20 minutes. For herpes virus, this takes 12 to 18 hours. But you start out with one virus particle and you can pop out 100 virus particles in less than a day. You can see how that would result in a lot of replication. So I won't do the maybe you guys answer questions for me, but what we know about evolution is having a large number of progeny and having a high mutation rate is going to create a scenario where you can get some natural selection and you can take out viral mutants. And so that's actually one of the fun things we get to do with viruses. Since it has a short replication cycle time, we can sort of watch evolution happening in real time in the lab. So what I study is protein-protein interactions that cause this fusion event, and also how the virus selects the cell that is going to infect. I use herpes simplex virus 1 and 2, which cause oral and genital herpes as a monovirus. So what we learn about those viruses applies to the other, well, there are 9 unique herpes viruses. I usually start this by telling you that most people are infected with 2. And once you have herpes, you have it forever. So it has co-evolved with humans to not cause problems for most of us. So it's generally not going to be a lethal scenario, but it's a ubiquitous family of viruses. And what we learned about herpes also applies to our understanding of other viruses that have envelopes, like SARS-CoV-2 or rabies or HIV or smallpox. There's a ton of them. The reason that I find interest studying this step of the virus replication cycle interesting is because if you can block the entry step, you don't have to worry about anything downstream. So we can learn about this to develop antiviral drugs. In addition, the proteins on the surface of the virus are the proteins that your immune system responds to. So understanding how those proteins work can help you design better vaccine candidates. And we do have, not me, but the world has some herpes vaccine candidates coming through human trials this year. It's very exciting. I have 2 graduate students and an undergraduate in my lab. You can use my name to find me on the back of the website if you want to talk about it.
Hello. I am Dr. Dean. Any of my Bio 191 students? Extra secret bonus points. That absolutely has nothing to do with your grade. I'll remember you're here. So these are some of the courses that I teach. Mostly general biology. Right now I'm teaching Bio 191. Many of you are in. As Dr. Kristriansky said, I'll be co-teaching with him Bio 193. Plants and animals. I'm the plant guy, he's the animal guy, which we usually end up being called at the end of the quarter. I have historically taught a plant biology course, but I haven't taught this one for a couple of years. But I also teach a plant physiology course that we do a lot of some biochemistry of plants, we use physiology, the lab-based component, we actually isolate some mutations that are related to metabolism. So that's kind of my more advanced course in my specialty. So my research interests, I love biochemistry. I don't know why, but traditional biochemistry, I just think it's fascinating. And I think the best biochemists on the planet are plants. I just am amazed at the kind of things that they make. And we've been able to exploit a lot of those, right, for human use and for human abuse as well. But I mean, if you think about things like nicotine, all the cocaine derivatives, opioids, caffeine, I mean, they make remarkable chemicals primarily for defense mechanisms. Plants don't care about us. They're making them to defend themselves primarily against insects. But a lot of those things are toxic. Just general toxins. So the plant has to be able to make them, and then they have to be able to somehow detoxify and store them. And that's where I'm really more interested in how they're detoxifying those and storing them for eventual use. And it turns out that a lot of times what happens is they'll stick a sugar on a molecule, the sugar makes it non-toxic, and then they transport it into the vacuole. So we're interested in the enzymes and the genes and the enzymes that are involved in making those enzymes that conjugate sugars to these molecules, and then the transport proteins that use that sugar as a handle and move those things into the vacuole. And the vacuole seems to be the main organelle where these things are stored. I spent a lot of time in the past 10, 15 years working on salicylic acid. Salicylic acid is the basis for the most successful over-the-counter drug in human history. Anybody know what it is? Aspirin, right? So plants don't care about aspirin. They make salicylic acid as a defense compound. It is a defense hormone. And it ends up being also kind of a general toxin and has some biological effects in plants. So we've worked out some of the enzymes. They simply take salicylic acid, they stick a sugar on it. We've kind of worked through those enzymes and identified them. And then once the sugar is added, we've been working on the transport processes, the biochemical processes that move them into the vacuum. I did that for a very long time, got bored with that, and then moved on to the next chemical that I'm currently kind of interested in. And these are flavonoids and anthocyanins. These are the red and blue pigments that you're going to see in plants. Generally, not always true, but most of the red and blue pigments that you see in plants are going to be anthocyanins. So the purple color of grapes, the red color of roses, those are all anthocyanins. And these are really easy to work with because you can visualize these compounds. Salicylic acid and radioisotopes is a little bit difficult to work with. These things have color, visual, you can see them. So you can generate a lot of mutations. So again, just a short end of the story here. We've identified some transporters that, again, this is a red pigment, and it's transported into the vacuole. We've been able to identify that particular gene and that protein that moves it into the vacuole. It's a real interesting group of proteins. They just directly use ATP and transport this thing in. But more importantly, we can generate mutations in the entire pathway of this particular pigment, and you can visualize it. So it's a phenotypic marker that you can just see the change very easily. So we've isolated a couple of mutants. This is the wild type. Here's the mutant that has a defect in this ability to transport it into the vacuole, and it's very easy to see. Right? We knew right away that we had a mutation because the color was different. We could then isolate the pigments and see which anthocyanin or which flavonoid was different. These are just different anthocyanins. You can see that they change. Some of them go up, some of them go down, but it gives it a different color. And that's the only defect, the only genetic defect between this one and this one is that this one doesn't have this transport protein to move it into the vacuole. So we knew this transport protein was important. So again, those are just some simple biochemical studies and genetic studies that we do using Arabidopsis, which is kind of the fruit fly of the plant world, I always say. Anyway, I think that's it for me.
Yeah, so,
couldn't make it today. He studies how cells integrate information and make decisions. He's our immunologist, and so he has these research areas. And so if you're interested in immunology, you might take one of his courses or reach out to him. Okay, with that, we'll introduce Dr. Gilbert. Okay. Okay. All right. So thank you. I'm Bill Gilliland, and there we go.
afternoon. I am the only insect biologist in the department. And this is my main insect that I study, which is the fruit fly, Drosophila melanogaster. It's very tiny. These are millimeter markers up here. So this female fly is only 3 millimeters end to end. And you can see that about half of her body is her abdomen. And most of her abdomen is made up by 2 very large ovaries. So she's a little machine for making eggs. And a single female can produce up to 300 eggs over her life. So these are creatures that have a very large reproductive capacity. And if you notice, sticking out of the end of her abdomen is this little whitish thing. That is an actual egg. So as she's been anesthetized with carbon dioxide, she's presenting this egg here. And so if you look at that egg more closely, here it is stained with DAPI, which makes DNA fluoresce. You can see this whole thing is about 0.5 millimeter long, 500 microns long, and this little tiny rectangle is what I am actually studying, which are the meiotic chromosomes. So I am specifically interested in meiosis, the process by which the genetic material is partitioned into gametes, which are your eggs and sperm. And so how do chromosomes get from parent to offspring? And so DePaul has an instrument called a confocal microscope. This is a real feather in the biology department's cap. For an undergraduate institution to have access to a confocal is a big deal. And students are able to use this as part of research programs. I'm actually the administrator for the instrument, so if you are working with a professor who wants to do confocal, you'll do training with me to get approval to use the instrument, and I can help you out with it. And so flies have 4 pairs of chromosomes, and they're big enough and distinct enough that we can make them out. So here are the 4th chromosomes, these are the X's, and then these are the 2nd and 3rd chromosomes. So we can actually see the chromosomes. And so my research focuses on what governs the movement of these chromosomes during the first meiotic division. You have 2 copies of each chromosome, one from mom, one from dad, and you need to pick one of them to put into the gamete. So how those chromosomes move away from each other. What are the genes that are required to make this work? If you break the genes that are needed to make this work, you get chromosome segregation errors, and that leads to the wrong number of chromosomes in the progeny. And that is the #1 cause of human birth defects. Aneuploidy causes things like Down syndrome, Turner syndrome, Klinefelter syndrome, and it is the #1 cause of human birth defects. Okay, so what genes are needed for this? How do they work? Okay. My research program, I am a geneticist by training, so I do a lot of genetic experiments where you do crosses and examine the progeny. I also do cytological work where we image cells on the microscopy both for DNA as well as fluorescent proteins like GFP or antibodies for immunofluorescence. I teach Gen Bio. I recognize a couple people from 191 here today, so thank you for making it. I also teach the sophomore genetics that is required for all majors. Me and Jessica Pammett are the 2 people who teach that. I've developed a new one of these CURE courses, these undergraduate research classes, called Advanced Genetic Analysis. I typed my own thing wrong. Advanced Genetic Analysis is the name of it. But it is Bio 362. This is a CURE course that is participating in a consortium called the Genomics Education Partnership. This is a group of about 200 faculty across the country, where a central lab at Washington University St. Louis generates genome sequence data, and then it is partitioned out to students in classes to annotate those genes. So as part of 362, you will generate new scientific knowledge. You will annotate genes in species that have never been annotated before. And as that data is collected and published in manuscripts, you will have the opportunity to be a co-author a couple years down the road after the class is over and your data gets published. As Tim mentioned, I'm also developing a new class that I'm hoping to run this spring. It hasn't been approved yet, but things look good for it, called the Science of Beekeeping. Like I said, I'm an insect biologist. I've mostly worked on flies, but I am moving into honeybees, Apis mellifera. And if you saw on your way in, I have brought in a demonstration hive, and I'll put it back out in the hallway after the meeting is over. So here I am in the DePaul Urban Garden, which is just down the street here, where I have 3 hives that I've set up as part of this project. And you can see we have a honeycomb here. You can see these cardboard-looking things. Those are brood cells. So those are actually baby bees that are in their pupal cases, and in a few days they will come out as adults. Okay. I'm not doing research on bees yet. I would like to add bees to my research portfolio, but the first thing I'm focusing on is developing this class. I want to mention the class, like I said, it should hopefully run in spring for the first time, and the plan is for it to be a non-majors service learning class that satisfies the science as a way of knowing distribution credit requirements. So you can, if you're a non-major, you can take this class and get your science credits for your distribution requirements. The first half of the class will be on the biology of the organism, so you'll learn what a colonial organism does differently than individual organisms like ourselves. And the second half will be learning the basics of working a beehive. Students will actually get the opportunity to come into the apiary, open the hives, take out the frames, do things like mite testing. You know, mites, Varroa destructor is a mite that has infected North American beekeepers over the last 30 years. And it's a real threat to colony health. So learning how to manage that is an important part of being a beekeeper. We're going to partner with a community garden in the Chicago area that wants to have a beehive. So the service learning component of the class is actually installing and maintaining a hive in the community garden to bring benefits to that community. Okay. Thank you for your attention. And our next presenter is Jing Kim.
Well, good afternoon, everyone. My name is Jing Kim. I'm faculty in the bio department focusing on physiology. And research-wise, I'm a reproductive biologist, and particularly focuses on molecular endocrinology. You can see several students from my lab here. So anyway, let's start with the courses I teach. I have taught a number of courses since I joined DePaul. Here you can see a pretty extensive list here, but the top 4 are the ones that I'm focusing right now. Mostly I teach human physiology, which is the course I created since I joined DePaul. I know many of you have taken this course before. I can see some of my previous students here. And another course I created here is called Mammalian Reproduction, which is a seminar-based course. And I see a past student here too. This course we could learn reproduction from all different angles, from molecular, cellular, and then relationship between central nervous system reproduction, steroid hormone receptors reproduction, like metabolism and reproduction. So it's like very thorough course, and it's very fun. We're going to study some current research through this course as well. I also teach biology capstone seminar and another course, which is a very popular course in the in the college, how the human body works. So these are the 4 courses I focus on, but you can see that I have taught a number of other physiology courses as well, which is kind of related to my background. I got my PhD in physiology from U of I, and during my 5-year PhD program, I taught all 5 years also physiology courses. So anyway, I'm glad to see some of my previous students here. If you haven't taken my courses yet, I look forward to seeing you in some of those courses in the future. Research-wise, as I said, I'm a reproductive biologist, and particularly we use molecular, cellular, physiological, a number of other approaches to study ovary development, and we're interested in hormonal signaling and also gene regulation in the ovary. And so you probably wonder why you want to study ovary, right? Just like many other researchers, we want to study different organs, different model systems. So the reason we are interested in the ovary is, as you know, the society is facing a lot of challenges like overpopulation, and also infertility has been a problem for many couples, right? 15% couples in the world are childless because of infertility. And also there are a number of diseases affecting human health, like ovarian cancer, polycystic ovary syndrome, or like infertility, and all these things can affect human health. So we try to find a way to understand the molecular mechanism underlying these diseases, and that will help us to find a better way to prevent and treat these kind of diseases or disorders, right? So currently, one of the signaling pathways we are focusing on is related to a very common nutrition molecule, which is vitamin A. So when we take vitamin A, it's going to be converted into a chemical called retinoic acid, which then bind to the receptor, come to the nucleus, and then modify gene expression. So my lab actually is one of the first labs actually demonstrate that retinoic acid exists in the ovary and can regulate ovary development. So currently, my lab is making conditional knockout animal models, and actually one of my graduate students just defended this summer, and she showed some very exciting data for one of the transgenic lines we made. So this is some very common techniques we use in my lab. We do a lot of animal work, we do dissections, treatments, tissue collections, we do cell cultures, we use histological, morphological studies, and of course we do a lot of molecular biological studies. And we also collaborate with others doing live cell imaging. Okay, so if you're interested in my lab, here's my contact info. Feel free to shoot me an email.
So Dr. Klosowski could join us today. She works both in the biology department and the neuroscience department, and she's currently chair of the neuroscience department. But she teaches biostatistics, Bio 375 Intro to Pharmacology, Bio 340 Behavioral Neuroscience, and Bio 342 Cognitive Neuroscience. And her research areas are in, as a behavioral neuroscientist, in developing animal models of brain injury, repeat concussion and subconcussion, sex differences with collaboration with Dr. Bell, and then the role of the APOE gene with Dr. Hrubeck. And she also studies neuroplasticity and brain injury in the survivors of domestic violence. So you can find all our faculty are listed on the Department of Biological Sciences webpage. So you can go there and find her contact information if you're interested in that type of research. Okay, that, one thing.
Hi everyone, I'm Dr. LaMontagne. I'm a biologist in the department. I also have an affiliate researcher appointment with the Morton Arboretum and an adjunct scientist at the Morton Wildlife Institute, Lincoln Park Zoo. So I study a bunch of trees and also do some urban stuff. I'm an ecologist. My degrees are all in ecology. I didn't write my universities here, but I'm also from the north. I'm also Canadian. This year I'm teaching ecology. I'm doing that class right now. In winter, I'm teaching introductory biology. And then I also teach some other courses like research methods and biostatistics. So people say, take stats and take it early. I wholeheartedly agree. That's the tool we use to understand things about biology in many fields. And I also sometimes teach population ecology. So I am a population ecologist, and generally I'm interested in how and why population patterns change over space and time. And those patterns related to plants and animals and other organisms. One of the things that I study is reproduction in trees. Just done a deployment use this week about acorn production. There's tons of acorns on lots of trees this year, so they're walking around looking for that. But I took a study on conifer trees, so this is white spruce. And on the, I don't know if I'm going to get this to work. Let's see. Yes, look at that. So these are new cones. So when you think about cones, right, you see them in the holidays, you have to think about brown cones. But they don't start like that. They actually start being green or purple and all these other amazing, really pretty colors. And during these large massed events, which happen once every few years, right, these trees reproduce en masse. There can be so many cones in the fall after those cones open up that we can see them from satellite imagery, which is a lot of cones. And you think that those are food for squirrels, birds, insects, and whatnot. A lot of the work that we do in the summer, in particular, is field work. So we go out and we count cones using binoculars, looking at the trees. We also have data sensors out in the field. So this is actually a temperature data logger that's being downloaded. So we bring computers out into the forest to do these things. Some of the work that I do, I have over 1,000 individually tagged trees up in Wisconsin, northern Michigan, and in Minnesota that we go and look at and understand their synchrony and why are different individuals doing different things. But then I use continental and global databases as well. And a few years ago, published a paper where we saw white spruce had, this is for 1 year data, had high reproduction in all of eastern North America, and it's low in all the west. And this affects bird migrations at these scales as well. So some really cool stuff. That's fun. And why might you care about this at all? Well, seeds are really important to future forests. So all of the little trees that come, that are going to show up are going to be influenced by seeds. They're food for birds, right? This is a bird that actually has a white spruce seed in its mouth. That's a pine siskin. And I work with people who study birds. And also I had a graduate student recently who looked at tick dynamics because you have lots of acorns, you have lots of small mammals, you have lots of deer, you have lots of hosts for ticks. So we also look at big spatial patterns and disease dynamics. Because again, I'm a population ecologist. It's just another population level thing going on. All right, climate change. Climate change is influencing everything. I work with an Oak Ridge National Lab project up in northern Minnesota where they build these huge environmental chambers to manipulate carbon dioxide and temperature, and there's full-size trees inside these chambers. So since 2017, we've been going and collecting data on that, trying to figure out both how is it influencing tree reproduction, but how is it influencing carbon allocation within trees as well. We also do some urban ecology work. We live in Chicago, and I think about urban areas not just as urban versus rural, but it's this mosaic of different habitats, right? There's a lot of variability. So we look at tree cavity availability, so they can either be made by woodpeckers, these perfectly round holes, or through decay. I have a grad student this year who's looking at red-headed woodpeckers. And how does acorn production, because they store them over winter, influence whether or not they stay over winter in this area. So lots of different projects. I have 3 graduate students at Technician and 4 undergrads in the lab right now. That's the stuff that we're working on. All right. Dr.
So I'm Dr. Shimada. Thank you. Right. So I am a faculty of biology and environmental science. I joined appointment. So half the time I teach for biology, half the time for environmental science. I'll show my classes in the next slide, but just to give an idea of what I do. So I'm a vertebrate paleontologist. So I study the ancient animals with backbones, and particularly my interest is on sharks, but I have extended my work and to include anything that occurs with a fossil sharks. So I have published papers on bony fishes, marine reptiles, and so on. I also have some publications on fossil birds as well as fossil mammals as well. But anyway, if in my lab I have 2 kind of pathways, people who are interested in anatomy, students will get a project with like comparable anatomy-based studies, and so we sometimes students will do shark dissections. I have some CT scan data of sharks, and they can analyze the skeletal anatomy of the sharks digitally. And another pathway is the kind of paleoecology or kind of vertebrate paleontology aspect. And most typical studies in my lab we do is to study what kind of animals lived during the age of dinosaurs. So here, this is like an image of how North America looked like during the age of dinosaurs, particularly the last part, the period called the Cretaceous period. And so you realize that these land masses, there were dinosaurs. But what I'm understanding is what was going on in an ocean inside. And so my field, I've been collecting fossils in Kansas, Nebraska, some in Colorado as well as in Texas. And I'll collect when I go to the field, like when I find some shark teeth, I get rolling out. I'll just bring a shovel and a scoop or a camera and take the sentinel samples back home lab, and if there's a student who is interested in working on a project, I'll just give them a dirt bag. See what you find. And so students will do like, we have a new student, MJ, just joined, but she just went through the acid treatment of rock like this, and then it takes time to dissolve the sediment, but then eventually what's going to happen is the calcium carbonate component that is that can be dissolved in the acid will go away, and then the residues, bones and teeth that are made of calcium phosphate, will be preserved. So here's an extreme case of like we have a lot of fish teeth that are on the microscope, and students will patiently pick, learn how to identify the species, and eventually catalog those specimens will be deposited in public institutions. And we publish every student project. It has a capability to be published in a peer-reviewed journal. So that's the goal. Anyway, I usually have about anywhere from 8 to 10 students at any given time. Right now, 3 graduate students, I think 3 or 4, I think 3 biology and 4 biology students and 2 environmental science students. I do. Because I have a joint appointment, I do need to set aside some space for the environmental science and study students, but I do also take biology students. Just so you know, I'm also the director of the biology graduate studies, so I coordinate the TA assignments and so forth. Also involved in the selection process of graduate students. If you have graduate students, by the way, just let me ask you this: how many of you are freshmen? Sophomore? Okay, junior? Senior? Okay, graduate student. Right, so this is a good mixture. But say if you are undergraduate students and you're interested in going to thinking about the graduate school, whether you apply here or not, it doesn't matter. If you have any questions about how does graduate school work, obviously it's good that, if you can interact with the current graduate students here, but also you can come to my office and then just have a chat. I can talk about how life graduate school works. Just a brief background, I'm from Japan. The thing is that I got my bachelor's and master's in geology and PhD in biology. So I actually know how graduate school works in different programs. It's remarkably uniform. All sciences have very common themes, so I can provide you a pretty good perspective of how graduate schools work. Anyway, let me go here. So here are my courses. As I said, I teach. I also belong to environmental science, so half the time I teach biology, half the time for geology. But for biology, I teach general biology too, especially evolution part. Gen Bio 2 is about ecology and evolution, and the first half is the evolution part. So I teach evolution biology part. Every other year, I try to teach comparative vertebrate anatomy class. This is we will look at the essentially anatomy, but very evolution perspective from fish all the way to mammals. The students will dissect a shark and a mink. It's very anxious because we only have 10 weeks, and then we actually go through the entire body system from both organisms, plus a couple of weeks of the skeletal anatomy, which is independent of the dissection part. So I'm currently teaching topics in paleobiology class. That's the cross-listed course, 345, Bio for 345, 445. And the other courses are environmental science, more geology-oriented classes, but I have had some biology who have taken some of my environmental science classes as well. So that's it. If you have any questions, just stop by. One thing, you can email me, but the easiest way, I always tell my students, don't bother making an appointment, just come to my office. It's much faster to talk rather than just going back for emails like when I'm available. I'm not available. So, anyway, thank you so much. All right, so.
Time with the BioSure. This is the new BioSure. So if you're a bio major, minor, maybe actually you can have a new shirt in the back. If you're not a bio major, we have some other shirts for you too. So if you're not a bio major, we have some other shirts you can look at. So as you already know, I'm Dr. Sparks. My research originally, I went out to study marine and freshwater biology, so I started off in that area. Started grad school in population ecology, then moved over into behavioral ecology. Studied a lot of things around sexual selection, mating-related variation, relation impacts, things like that. And then when I came to Chicago, I found a little system that I've always been fascinated by, which are these parasites that are what called zombifying parasites. Parasites that get into a host and then change their neurobiology, their behavior, everything about them in ways that make them more attractive to the predators. So the classic example you might hear about are rats and mice that get infected by T. gondii. Instead of running away from cats, they run towards cats because the parasite needs to get into that catfish life cycle. Hyenas that are shown to move towards lions, things like this. Monarch exotic hyenas and lions, it's a crustacean fish, it's local. So I've done a lot of work locally in Kildare, which I told you, I think it's Kildare area, right? There you go. So that's local. It's become kind of a very well-known system because of all the work we've done locally. Lots of undergrad-based and then grad-based research students coming and going through the projects, and I do enough variation in types of questions that usually interest students have to match with the kinds of questions, skill sets they want to develop, kind of thing that they might want to do afterwards. Even though I do field-based ecology, I have lots of pre-med students working in my lab, one med school, and those kinds of programs, and the kind of skill set they develop, often students publish projects, things like that in the lab. More recently, I've got a couple of students here on courses, we get involved in microplastics because we have a field class in marine biology we teach in South Carolina, which I've taught with Dr. Kashmansky, and they're everywhere, right? They're everywhere, so it's become a theme in that part of the research, at least the course-based part of the research. That's resulted in posters and working on potential publications and things like that around that subject. I teach a bunch of classes, some of them new classes, Bio 101 if you're first year. I see some of my Bio 101 students here right now. This is a brand new course we put in place. It's all about helping students transition into college and get all the things down that we all want to know about, maybe don't know about. And Capstone students give advice on things they wish they'd known. A lot of these things are now in that class. That transition to college, that's a new one to start this quarter. I also teach marine bio for non-majors, 192, so I do the eco and the evo and all, depending on who I'm teaching with. Animal behavior, I've done teaching for a few years, so I teach that. Aquatic biology, I've done for teaching for years either, but I teach this 318 course in South Carolina in December. So a lot of people know December into session, you do an entire 10 weeks basically in 10 days, and then some time in the winter quarter. It's full immersion. It's fair to say that to the people, right? It's a full immersion experience. We live together, you're up early, you work until late, you get a huge amount of work done in a short amount of time. It's kind of a sometimes transformational experience. Many of these, many of these kind of immersion style courses can often be transformational. It's just what you get from them in terms of the scientific development. We got that, and I do capstone, and then a behavioral hierarchy class. I've looked at that area last few years quite a bit. And again, often undergrads, often grad students, depending on their interests, all those things are possible inside the research program. And that's just a thing from South Carolina. Little fish we pulled out on the side. All right. Thank you much. Thank you all.
We're going to present some of our lab coordinators and our teaching faculty. And so Rima Markauskas couldn't be here today, unfortunately, but she is involved in the general biology sequence in general biology, non-majors course, Bio 125, 201, human anatomy, which is a popular course, 202, human physiology, and then she has a 302 course. She is lab coordinator for cell biology and genetics, and her research experience, her background is in aquatic physiology and stress tolerance. So her contact information is here, and it is also on our faculty and staff webpage. Claire Lorenz is, you're very,
Hi, everybody. Sorry if my voice is a little hoarse, I've got a little bit of a cold. Let's see, I just want to quickly introduce myself. There's a lot of Bio 191 students here, so I'm one of the lab coordinators. It's kind of my primary role, so I've been coordinating on sequence 191 right now, and I also do off sequence 191 winter quarter, and then in the spring quarter, I get to do Bio 192. So hopefully I'll get the chance to meet a lot of the labs. I've also started lecturing for Bio 192, the ecology portion. I did that for the first time this past spring. had a great time. It was a lot of fun. And I get to do that again actually twice this year, so I'll be lecturing one into winter and spring quarter. So hopefully I'll get to see a lot of you in that class. And so my research background, I got my master's here at DePaul a number of years ago now. So my focus was species diversity and ecosystem restoration. So you can actually see these photos in the main DePaul atrium in the north building. So I've got some photos of microarthropods. So I looked at how above ground plant diversity affected the microarthroid diversity in the soil. So we looked at different levels of restoration ecology in Chicago wilderness. So if that is interesting to you, I'd love to talk to you about that. And I also added a few kind of fun pictures. My kiddos up there. I've got a 2-year-old and a 5-year-old, so they are troublemakers right now. And then my other 2 troublemakers, my kids. All right, so I hope to see a lot of you in lab. And let's see.
Thank you. Betsy couldn't be here today. She's also a lab coordinator and instructor. Her office is in McGowan North, room 113, and her areas are microbiology and biotechnology. She is involved in our general biology sequence in General Biology 1, so some of you might have her as your instructor there. She does Bio 161, another general biology class, Bio 210 and Bio 370. And her background is in immunology, like Dr. Funk. So if you're interested in immunology, there's a couple people to speak to there, and she is on our faculty staff page. And we have our teaching faculty, so Rick, Dr. Rick Hudson, Dr. Terry Fitzpatrick, and Dr. Sarah Richardson. I thought I saw Dr. Hudson. Oh, yeah, and Richardson. Do you want to come up and say something about yourself? Very humble, Dr. Hudson. Don't want to say anything?
No, that's fine. I'm just looking forward to meeting students that will be teaching in 1992 next quarter. So I'll see some of them.
So thank you. Thank you. So they're on our faculty and staff page as well. So you can find them there and email them if you want to talk a little bit more with them. Dr. Elizabeth Piment couldn't be here either. She teaches Bio 155, Introduction to Biology, Laboratory, the Science Behind Human Health, Introduction to Biology for Non-Majors, Genetics, so she is the other instructor for Genetics, and Cloning and Biotechnology. And we have Dr. Kate Soderstrom, who could not be here either. And so she teaches a number of courses for us, including brain and behavior, and general biology for majors, and biostatistics. Okay. Unfortunately, at the last minute, Sarah Fink, who's our academic advisor, had to cancel. She wasn't feeling well today. So her contact information is here, but her role is to help students better understand their degree requirements. So the faculty are generally better about what is this career about, what is the graduate school like, what are some possibilities in terms of my profession. Sarah is the person to talk to about the specifics in your classes at DePaul, the requirements, things like that. Has a lot of experience in that area. She's worked at DePaul since 2018. She's with us more recently, but she's a double demon. And you can make appointments through Blue Star. That system, faculty love Blue Star. I mean, I'm not serious. It's a new system, but hopefully it works better for students. We complain a lot about, it's a new system, so when there's something new also in fitness, it tends not to work that well. And so she has some, she has a Zoom meeting on navigating degree progress report that is coming up. James Roth is our departmental assistant. He is not here. He is in the office with the General Biology office in McGowan North. I just want to show you his picture. I'm going to offend a bunch of people, but probably the single most important person in the department. Person knows how everything works.
Just say that probably.
It's a very important person. Yeah, so graduate students, I'm sure, would not be offended by cookies and things like that. So, but as a graduate student, we rely on our departmental assistant very much. And our departmental assistant here is much better than my PhD program. So our departmental assistant had a jar that said, ashes of trouble students. So they were generally pretty helpful. Graduate students. So we have a graduate program that I think is really a great graduate program that does a good job of training our students. We've invested a lot of resources in trying to make it a strong program. So we have a master's in science program that is active, and your graduate students are doing original research projects. And there are also teaching assistants in your labs. So when you interact with your teaching assistants, know that they're doing this extraordinary work of both teaching you the lab components of classes, and then carrying out an original scientific research project that in many cases is going to be published in an original peer-reviewed journal. So it's a huge deal. So very important members of our department. I'm going to ask them to stand up. And so you can see them.
Now let's give them a big hand.
Yeah, not that the faculty and staff don't work hard, undergraduates don't work hard, but our graduate students are incredibly hardworking. So that round of applause is very well deserved. And we have student organizations. So we have several of them that are associated with biology. We also have a neuroscience club that many biology students are active with. There's a predental club, DEEP, DePaul Ecology, Evolution, Physiology course, pre-vet, conservation club, which is a new club, and Doctors Without Borders. So are any of those reps here by chance? I want to say something. What is your club?
Hi, I'm Campbell. I'm current former president of DEEP. We're trying to get the club going again. We currently have someone who is looking into taking the president position, but we're looking for a treasurer to help handle funding through OSI and try to create new opportunities. So DEEP is the Ecology, Evolution, Physiology Club. We are a professional club that tries to connect students with lab opportunities, research opportunities, and other events that are related to our nice little acronym. So it's a good way to meet people and engage with new opportunities, both inside and outside the club.
Thank you. With the Conservation Club, they're also looking for students interested in joining the leadership. These clubs right now in a little bit of a transitionary phase. We do have pre-med pals. Are you here? What was that? Did you want to speak?
Hi, I'm Shayna. I'm an art major here, so I feel kind of like an imposter right now. But I am currently slated to be leading DEEP as we head into this year, and like what was just said, we're looking for a treasurer/secretary sort of position right now, and once we find somebody to fill that slot, then we'll be able to get the club re-registered, and if anybody's interested in any of that, ecology, evolution, physiology, learning about Chicago, learning about Chicago's urban environment, species interaction, anything along those lines, please let me know or Dr. LaMontagne or anybody, and we will get you some more information. I'm hoping that it's going to be a good and fun year as long as we get the club registered.
And so you get to learn a lot of things and do a lot of fun activities with the clubs. But it's also a great form of peer learning. The contacts you make as students with each other, often some of them are going to be contacts you have for the rest of your life. So you can form very strong bonds in these types of settings. Pre-med pals, it's a rep here. I don't think they could come at the end, but you have a slide that they send. They're a new club, and it's probably pretty self-explanatory. So if you're interested in pre-medicine, in medicine, this is a club you want to check out. And they have a QR code here. So if any of you want to get that information, you can also do that.
1 second, 2 seconds.
Got it. Great. Okay. There's groups. There's a lot of students at DePaul are very active in many ways, and so there are groups
that give you the opportunity to have some focus activity. So we have a group called Diversity in STEM for CACH students. That is an inclusive group for all races to share ideas, opportunities, and connections with Chicago communities. We have the Society for the Advancement of Chicanos and Native Americans in Science. This has been a very active club in the past. There's a national conference that you can attend if you're part of this. We have LGBTQ out in STEM, so there's a system that empowers LGBTQ students in STEM to succeed personally and professionally through connection, leadership, and advocacy. And we have Ask Women in STEM, so fosters sisterhood, achievement, and philanthropy, and promotes women in STEM leadership, friendship, and support. So a lot of great groups that you can look into. We have student support services. So the Office of Multicultural Student Success, the Dean of Students Office, University Counseling Services, and the Vincentian Assistance Fund are all places that provide critically important services for students. So it's one of the things I'll give you a piece of advice is, as a student, take advantage of the resources that exist. Universities often go to great lengths to try to provide resources. And often, when you leave, it's one of the things you can regret is, I should have done more of this and taken advantage of some of these resources. So we also have research opportunities. There's a Chicago area undergraduate research symposium, CARS. This happens annually here in the city of Chicago. It's hosted by different institutions every year, so this is a chance if you're doing research to sign up and meet others doing research and give a presentation. These experiences are incredibly valuable and very important sources for it. There is the STEM Undergraduate Research Showcase and LSAM that are also important opportunities for you to give presentations and showcase student research. LSAM also has funding for students to do research. There's social media, a little bit outdated. I didn't change this, but these should be mostly active. And yeah, and then final thing is we have a monthly seminar series. So we're going to have a Journey seminar October 13th. Relatively new hire in environmental sciences, Dr. Noé de la Sancha is going to talk about his journey into becoming a researcher and a university professor. And his specialty is in mammal systematics and urban evolution. Urban evolution. And we have a talk from a researcher at Northwestern University that's going to showcase their biotechnology graduate program. So that will be November 3rd. And then we have our Dharma Day. We don't have a set talk yet in January. That's coming. But our Dharma Day seminar will be given to our new hire Dr. But that's going to be February 16th. So some exciting seminars coming in the near future. Okay, with that, we will open it up. So like Dr. Sparks said, there's some t-shirts. So those can be handed out to the biology students. And then there's going to be refreshments outside, an opportunity for you to faculty hang out for a little while and talk to you. And so hopefully you'll take advantage of it. Thank you everyone for coming. It was really nice to see you.
Graduate students, if you can sign in, make sure
there's a bee display. Dr. Gillen has live honeybees, so do not open those, but you can enjoy them safely from the case.
Yeah.